Yes, sir. Good morning, sir.
Good morning. Am I audible? Yes, sir.
Yes, sir.
Yes, sir. Good morning, sir.
We can't hear you, sir.
Am I audible, please?
Yes, now you are audible.
Good morning. So, shall I start? Welcome to the third day of the presentation, third day of the training program. Today is also a concluding day. Today at the beginning we have with us Dr. Subhashi Otta from ICR. He is a principal scientist at ICR Siva, Chennai, in the field of skin diseases, their diagnosis, pathogenesis, and control. His major focus on the control of the viral diseases like WSV, HPLV, through RNA silencing, etc. May I request Dr. Otta to have a presentation? Thank you, sir. Dr. Otta, please. Yes, sir. Hello, sir. Yes, sir. You have to present now. You have to put something as your entire screen, window, or tab. No, now present now, then it will come where from to present. The drive it will show which where from it will present. Is it coming? Your presentation is coming?
No, sir. The three options I'm getting. Your entire screen, a window and a tab.
Window में क्या? कहाँ से? From where you want to present?
It's here in my front desk.
What's that?
Is there in my screen also? I have opened the PPT.
Okay, but it is not coming. It is not coming in the screen. Just present now, then go to your folder where it is.
It's okay now, sir?
No, now it is coming. Yes, it has come. Yes. Yeah, it has come. Continue, sir.
Hello, sir. Okay, sir.
Continue. Sir, not coming.
Hello, sir.
Again.
Yes, something, no problem. 1 minute, sir. I'm checking.
No, how to do? Past present now? Okay. Sir, now, present now, then share your entire screen. After that, okay. Then okay. Then open the folder and go to the presentation. Then as it is, normal. As it's seen result. Yes. Yes. Sir, just open the PPT. No, don't stop. Open the PPT. Yes. Yes, it has come.
Is it coming?
It is coming. But if there is some problem in the slide, then go to the type.
Okay, sir.
Click the hide button. Click the hide button at the bottom.
Okay.
The bottom. After stop sharing, there is a hide button. Click there.
Okay.
Yeah. At the bottom. Yeah.
Okay, sir.
Okay, sir.
A very good morning to all of you. Very good morning to all of you. At the outset, I thank the organizer, particularly Dr. Manna, for giving me this opportunity, and also the director of CIFRI to discuss about an important topic, that is the viral diseases of stream. And you know, viruses are very virulent and they can bring high mortality. And next step, how to manage this pathogens.
I'm not listening.
So, before going to the main topic, let's discuss a little bit about, in general, about the disease. So, you know, there are three factors. One is the host or your shrimp, and which you are going to culture, and the next is the environment. There's the pond water where you are culturing the shrimp, and the pathogen, the agents which cause disease. So, there is a very delicate balance between these three. So, if there is any little bit of imbalance in any of these factors, then there will be outbreak of disease. And how this imbalance occurs? For example, host. If the immune system is low, then there is a chance that pathogen will invade and cause disease. Similarly, your environment, if it's not suitable, for example, if oxygen is low or your toxic gases like ammonia, nitrite, or hydrogen sulfide, they are very high. So these all together, this will bring stress on animals and it will bring down its immune system. Now, pathogens, suppose they get any suitable environment or particular conditions, so their number will increase. So they will exceed in number then, and they will disturb the diversity, and at that time they will be able to cause disease. So that's how the balance breaks and disease occurs. Now, why we are getting very often very new and emerging aquatic animal diseases? So the first factor that because our intention is to increase the aquaculture production. So to do that, we have to bring the aquatic animals from somewhere else, or you have to send from our place to some other place, depending on the demand. So because of this translocation, when you are transporting or you are importing the animals, along with that also pathogens will be there, because we cannot separate the pathogen from the animals. So in that way, pathogen may spread from one place to the other. Second thing, so when you are planning to go for a higher production, definitely you have to increase the diversity. We have to search for new species, how you can culture this, and which are having very fast growth rate. So to get better production, so whether it's finfish or crustaceans or molluscan, so we find new species to introduce into the system. So in that way also, we are able to increase the disease chance. Then new production approaches. Suppose the area is limited, population is increasing, we don't have much area to do the culture practice. So we'll try to how best we can utilize the same area. For example, multi-trophic aquaculture, all the three layers, the top layer and the middle layer and bottom layer, everything will put some species depending on their niche, and that's how. So there will be definitely pressure on the system. So that may result in the outbreak of disease. And the last thing is, so by day, so new things are coming. So we are improving our diagnostic procedures to make it more sensitive and more specific. And also we are regularly doing the monitoring or the surveillance. So the meaning that more you look, so more you find. Because you are taking more effort to do the surveillance and you to improve diagnostic methods. So maybe for that reason we are finding new pathogens, which you are not used to do earlier. So stress is one of the most important factor which causes disease. So it interferes with the immune system, the immune cells and all. So that's how it brings down the resistance capacity. It can fight against the disease. So once the immune system is down, then the pathogen gets opportunity, because it cannot be destroyed by the immune system. So it can infect and it can multiply and it can cause different type of diseases. Now, how the stress comes? Starting from the beginning, like from the hatchery system. So if the seeds are not of good quality, if they are poor in quality and they are weak in, they cannot they are not active. So that way there will be also stress on the animals. Second thing, when from hatchery, when you are doing the packing, how you are doing the packing, whether sufficient oxygen is given or not, whether the temperature is maintained during transport, and how far you are transporting. Based on that, you will have stress on the animals. The third thing is when you are stocking in a pond. So definitely, if you compare a hatchery condition where everything is optimal, so everything is a luxury for the animals. your say oxygen, your temperature, and other environment, everything is optimal. But when you are talking, everything is just opposite. So nothing is in your control. Everything is beyond your control. So that will bring severe stress because suddenly the animals are going from one environment to the other environment. Then fourth thing is density. So every animal requires some space. So if they're going beyond the density, then definitely there will be stress on animals. There's a limit to how much stocking density you should have. And the last thing is the management practice. How best you can manage the system, like how you are managing the aeration system, if you are putting sufficient aeration or not, how you are managing your feed, like through the check prayer and other things to handle, how you are managing the water quality, whether you are maintaining the best environment. Based on all these things, then there will be, it depends how much stress it will bring on the animal. Now, when you consider the stream diseases, so all kinds of microorganisms have been responsible to cause some of the other diseases in stream, whether it is virus, bacteria, fungi, or parasite. Everything have been detected in stream. If you consider virus, so far more than 25 viruses have been reported. And if you see the trend during the past few years, every year you find one new virus which has been reported. So virus is generally considered that they're highly virulent and then bring severe mortality, and that's how they could bring huge loss to the farmers. So farmers are more vulnerable to virus. The second is bacteria. Earlier it was thought that bacteria is not that important. Some disease it may occur, but somehow if you put some sanitizers, antibiotics, somehow you can control this. But if you see the present scenario, where the early mortality syndrome or acute hepatopancreatic necrosis is HBND. So if you see that bacteria, some bacteria like Vibrio parahymicus and some other species, they can harbor a specific gene or plasmid, and that can be more severe than the virus also. That can also bring mass mortality. So this also you cannot ignore. Fungi, if you consider, so generally so far they have not been that much important. Only in hatchery conditions at larval stages, some of the fungi they cause problem and all, but usually it has not gained that much importance. And coming to parasites, some protozoan parasites and other particularly microsporidians and all, you see that earlier parasites were not considered that much important, but at present scenario, the parasite called EHP or enterocytosis hepatopenia, that has been gained the prime position. Farmers are more worried about that EHP than the virus or bacteria. So time and again, the virulence factor changes, and pathogens which are not earlier found to be that much virulent, they become virulent and they gain that much importance. And we think how to control this. Coming back to our virus, so based on nucleic acid, so you can divide into two major types. One is DNA virus, another is RNA virus. So in DNA virus, you can find different types. Say, for example, parvovirus. So it includes the main virus that is IHNB, infectious hypodermal hematopoietic necrosis virus, and also like HPV, SMV, and LPV. All these viruses are included under parvovirus. The second group is baculovirus. Earlier, you know, MBV or monodon baculovirus. It was very important in case of penis monodon because it was to reduce the growth and bring severe economic loss. But nowadays, in Banama, it is not that important. Other virus, baculovirus pneumoniae or baculovirus mid-growth gland necrosis and TCBV, PHR. So many viruses have been reported in different times, which comes under baculovirus. Then one of the most important virus is white spot syndrome virus, which is totally put in a separate group, the single virus. So it belongs to Wispo virus and the family Pneumoviridae. And in different time points, this has been named in different types. Like earlier it was called as MBV, systemic ectodermal and mesodermal baculovirus, or RBPJ, Penis japonicus rod-shaped virus, and WSBV or white spot baculovirus. Like so many names it has got. but finally it is called as white spot syndrome virus, WSSB. And also there are iridovirus, and iridovirus have been reported earlier, but that is not that important. But present scenario, if you see, one of the iridovirus, decapod iridescent virus 1, DIV1, that is again getting important now because it is seems to be little virulent and kills the shrimps, though it is not found in India. But still, many farmers now worried about this virus. Coming to the RNA virus, so the first one is picorna virus, to which Torah syndrome virus belongs. This again, it is an important virus, highly virulent virus. It can cause mortality, but so far in India we have not detected this virus. And rheo-like viruses also it has been reported in the shrimp, but somehow it has not gained that much importance. We have not recorded any mortality or other aspects regarding this virus. Then also Toga-like virus, lymphoid organ localization virus, that also was reported. Rhabdovirus, that has been detected, and yellow-head virus. This again another virulent virus which is capable of causing mortality to shrimps. So that is a separate group. And subsequently, based on the report, there are other groups also. One is Totoviridae, to which the AINP or infectious myocrosis virus belongs. This also, it was highly virulent when it was reported in Brazil. And now, for a long time, it was not detected in India. But recently, in 2016, it was reported from India. And from since then, in many places, many reports have come. And last one is Noda virus. So another one is Penis banami Noda virus, PVRD. And another is covert mortality Noda virus. Among these, CMNV is again important in China. And it was reported because somehow in India it was not reported. If you see the OI listed, OI is the institute which lists the pathogens based on their virulence and how they spread, how they bring economic loss. So based on that, if you consider shrimp viruses, 5 viruses have been reported by OI, which has been listed in OI. So these are white spot syndrome virus, infectious hypodermal necrosis virus IHHNV, Torah syndrome virus TSB, yellowhead virus YHB, and infectious myocrosis virus. So it should be know about these 5 viruses because definitely as they are listed, they are important. So why virus diseases are so important? Because now they are highly virulent, they can cause severe mortality and they bring severe economic loss. So if you see the older report under the white spot virus, TSB, YHB, or IHHNV. IHHNV in the beginning stage, it was highly virulent and particularly to Penis stylosis, which was able to cause 100% mortality, but subsequently its virulence has come down. It's not that important, it can cause mortality, but it can cause severe deformities, that's why it is important. So in any way, some other way they cause economic losses. You can see billions of dollars are lost because of this. And if you see a very recent survey from our institute, so white spot syndrome virus alone, it was responsible to cause $238.33 million during 2018-19. This is also again only in 3 different states: Andhra Pradesh, Tamil Nadu, and Gujarat. So if you take a survey in entire India, definitely it will be much more than that. So now also white spot syndrome virus brings so much loss to shrimp farming industry. If you see the different the world map, in different time points, different viruses, they have originated from different places, but they are not limited to that place through transportation or some way, whether by the live cultured animal or the live shit. So they have been transported to different places and they have spread to different parts of world. And now all the viruses you can find in many of the places. If you compare the viruses based on the size, there is a wide variation. You can see here the largest virus, white spot syndrome virus, even this is called as the largest animal virus, and the smallest virus, is the IHH, which is only 20 nanometer. Similarly, another peculiarity in shrimp is that you can get multiple viruses in the same host. So like you can get MBV, HPV, WSSB, or even if you consider Vanamai, you can get WSSB, YHV, IHNB. So 3 or 4 viruses or even more than that, together they can be in the same host and they can cause problem. Coming back to know about each individual virus, the first is white spot syndrome virus, which is called white spot virus disease. And you know this is the highly virulent virus, even though it has been 30 years from its first report. Still, it is highly dangerous, highly virulent, and now also it can cause the same 100% mortality if it infects the shrimp. There are several clinical signs, and the major clinical signs you can see white circular patches on the body surface, and you can see everywhere in the place. So it is caused by a large DNA virus, double-stranded DNA virus, belongs to Histovirus, family Nimiviridae, and you can find in all the 3 water systems: the freshwater, brackish water, or marine water. So like this picture, you can see that white circular patches on the surface. If it is Penis monodon, it is very clear in the naked eye. You can see in the shrimp, in the carapace, and also in the entire body. It's another picture. It's another picture, as you can see from the carapace and in the abdomen. Everywhere you can find the circular white spots, and it's very easy to identify. You can make that WSSB is present. And Penis marmai is called as Pacific white shrimp. Because it's white in here, it's difficult to see the white patches. But if you will lift the carapace and dry it little bit time, 5-10 seconds, then this white patches also will be very clear. Another sign is in Vanamai, it becomes reddish discoloration. Once you see the reddish discoloration, you can just guess that white spot syndrome virus, maybe they are in the shrimp. So it has a very wide range. As I told, it can occur in freshwater, in brackish water, and marine water, in the freshwater prawn, marine prawn, and all kinds of decapods. They have been completely infected by this virus. Some act as a host and some act as a carrier. So because of this, it's almost like bacteria. So once it comes to the system, it's very difficult to eradicate the virus. So best way to keep it away, not allow it to enter to your system. Second thing is infectious hypodermal and hematopoietic necrosis virus. And earlier, as I told, when it was first reported in Penis stylorostris, that time only it was highly virulent. It was causing even mass mortality. But subsequently, its virulence has come down. But one major thing is does the clinical sense, the Ront deformity syndrome, RDS. With many other deformities in the body, in the antenna, in the rostrum and the antenna. So because of that, the appearance becomes bad and it doesn't fetch much price. So again, it's a smallest DNA virus belongs to Parvoviridae. That 4 different genotypes are available. Again, like white spot virus also, though it doesn't have many carrier and other things and done, but it has been reported in many of the majority systems. So next is yellowhead disease. So far, it has not been reported from India. Only long back in 94 around, there was a single report that was based on histopathology analysis. However, after that, it has not been reported from anywhere else in India. And so that's why you can consider that it is absent in India. Again, it will have almost all the virus disease, you see the same clinical signs. But the most prominent clinical sign is the yellow color. It starts from the gill, spreads to the carapace, and then spread to the entire body. So it's a corona-like RNA virus, belongs to genus Oca and family Rhomaviridae. And there are 6 genotypes, only one genotype is virulent. You can see the gill sandal, how this becomes yellow color. gills, if you open the carapace, totally it becomes yellow color. Spread to carapaces, then the entire body becomes yellow in color. That's why it is called as, that starts with the head, head becomes yellow. That's why it is called the yellow head virus. Again, it has wide host range. Many strains and they are affected by this. But about carrier status, it's not much known. Tora syndrome virus, it is called so because it is first reported near to a river called as Tora. Based on that, it was called a Tora syndrome virus. And it also a highly virulent virus and it can cause mortality in shrimps. The major clinical signs are black spots on the body, entire body, and the tail flap. It is red in color, and the leaves, and in the entire body. Particularly the tail region is reddish in color, and this will particular serration like this. So these are some of the typical clinical signs. You can guess that the shrimp is infected by Tora syndrome virus. This again some pictures. Again, a wide variety of strains, almost all penguin strains that are affected by this virus that is reported either experimentally or naturally. The strains get infected by Tora syndrome. So this shows that this has a potential to infect many shrimps. And the last is infectious myonecrosis virus. So far, like for a long time, it was not reported from India. But as I told, from 2016 onwards, this found was first reported from West Bengal, and now it has spread to other parts of the country, like Andhra, Tamil Nadu, Gujarat. Also, many places it has been detected. And the main clinical signs from the tail region, it starts with the necrosis, white color sepsis-like things, and sometimes this part also becomes out like the shrimps totally lose this part, and that's why the market value comes down. And though it is virulent, it can cause mortality. But nowadays, the virulence has come down. Maximum 30 to 40% mortality you can find. But still, it is important. And its RNA virus belongs to Cotiviridae. So these are the clinical signs of from the tail region, how it starts, the reddish discoloration. And then it spreads to abdomen slowly. Some more clinical signs. And it has limited hosts like Vanamai, Stylorhistris, and Monodon. So from other systems, so far it has not been reported. There are some other viruses which have just been reported, but how much economical loss they cause, what is the characteristics of the virus, it is not known. So one of that is abdominal segment deformity disease caused by a virus. So this was reported by Flazel group in Thailand. So they found that all these shrimps, they carry a virus, and they were able to reproduce the disease also, but further characterization or other research activities has not yet been done. This is another virus called as Lame-Sing virus, which causes monodon slow growth syndrome. It has been reported only in monodon. So that time when monodon culture was there, it was found to be important because it considerably reduced the size and weight of the animals. That's why it was a loss for the farmers. And it has several clinical signs, but it's not that important in Vietnam. Another is called as loose cell syndrome, which was reported from our institute, and they reported that it is called by a virus-like particle. But as I told in other cases here also, this virus, what is this virus? It has not been characterized, and the other things has not been. So that's why it's not considered as that important like the 5 listed viral pathogens. Now recently some more viruses have emerged. One is covert mortality Noda virus or CMNV, this is the RNA virus. And as you can see, the main part here is hepatopancreas. So earlier it was confused with early mortality syndrome. The thing that so maybe because of EMS this happens, but when it's analyzed, it was found that because of a virus, this disease comes. So they named it covert mortality Noda virus. And the latest virus is Decapod iridescent virus. Initially, it was called as shrimp hemocyte iridescent virus, and they found that only in finite shrimps occurs, and when the particular case is infected. But subsequently, they found that it can also affect the freshwater prawn like Macrobacterium rosenbergi. It can also affect the crawfish, so many other fish, and so that's why it was further in the taxonomic body they change the name to decapod iridescent virus because it affects many decapods. So far, this has not been reported from India. So then coming to the diagnosis of this virus disease, diagnosis is an important part, which also a part of management practice. You can decide about the culture practice. So microscopy methods like light microscopy or electron microscopy, histopathology, then different protein-based methods like ELISA or polyclonal monoclonal antibodies, then DNA-based methods, DNA probes, and or PCR, RNA-based methods like RT-PCR. All these methods have been well established to diagnose the virus. Sometimes for earlier for the light microscopy under to detect NBV under occlusion bodies, and it was very helpful. So from the field side itself, you can decide whether the larvae is infected by virus or not. So now to do the diagnosis, the one step is sampling. That is very important. How you do the sampling to do the diagnosis. If you consider histopathology, so you know there are different fixatives, but for shrimp, Davidson fixative is considered the most suitable. That's how it keeps the tissues intact, and you can diagnose the disease. So in shrimp, the hepatopancreas is the major part which undergo deterioration very quickly as soon as the animal dies. So if you are considering for histopathology, you should consider only moribund shrimps or very freshly that immediately some fish has died, shrimp has died, that you can consider. Little bit delay means it's not suitable for diagnosis. So as soon as you get the shrimp, so both sides of the hepatopancreas you have to inject. So first you have to fix the hepatopancreas because that is an important organ which undergoes deterioration because as it contains a number of enzymes. Then abdominal middle part and the last part inject. After that, depending on the size of the shrimps, so you can cut out the exoskeleton and put in the fixative. So after 48 to 78 hours, you can go for processing. You can process the tissues, cut the sections, then do the staining and observe under microscope. Similarly, if you have to do electron microscopy, you can fix in 2.5% butyryl gel and then subsequently process for microscopy. For molecular biology, whether you are going for DNA virus or RNA virus, so depending on that, you can fix in alcohol or RNA lighter. And sometimes, if you want to test directly, you can also put in dry ice. Or if you are getting the samples from far place, you can put it in dry ice, transport to lab, and then you can process it further. And you can extract a DNA or RNA. Depending on what you want to do the PCR or real-time PCR, or you want to do sequencing, any aspects you can analyze. So this is coming back to histopathology. You see, histopathologically, each virus, they'll have a particular pattern. So that they found inclusion bodies or occlusion bodies, in which part of the body, whether it's gill or lymphoid organ or hepatopancreas. So they'll have a definitive structure. So if you consider these two DNA virus, WSSB and IHS, you see the types of inclusion body they form. Here in IHS, you found the inclusion body and surrounding that a clear area, which is not in case of white spot syndrome virus. So that based on the typical structure and based on your experience, through histopathology, of course, histopathology is considered as a golden standard because getting false positive will be very less. But rather than that, you should have the greater degree of infection, then only you will be able to detect the virus. Similarly, in YHV, TSV, everything, all the virus, they have a definite pattern and different types of inclusion bodies. So you have, by little bit of practice and your experience, you can detect the virus. Molecular methods, a number of methods are available, whether single step PCR or nested PCR or a single tube nested PCR. You can detect simultaneously 3, 4, or 5 pathogens through multiplex PCR. You can quantify how many virus particles are there in the stream to a particular amount of tissue by real-time PCR. And the most recent method is a CRISPR-Cas-based method, which is more specific and accurate. Different diagnostic methods, slowly it is coming up based on this method. So single step PCR you can do, or multi-step is like the DNA virus or RNA virus. At time 2-3 viruses you can detect. You can also, if you don't have access to sophisticated equipments like thermocycler and all, centrifuge, so you can do the loop mediated isothermal amplification just with the help of a water bath and some reagents through the color development. You can also get very accurate result. It has been standardized for many of the stream viruses. Or you want to go still higher stages, you can do microarray. This is particularly important for development of SPF stocks and all. Some latent virus handle to detect. Or you can do the sequencing, you can target a particular gene, or you can do whole genome sequencing. Like this for white spot syndrome virus, so different genomes are available. China, Thailand, or Taiwan, so different genomes, and you can do the further analysis, how you can characterize the virulence characteristics. So based on the research need, you can decide what kind of diagnostic methods you can follow. Protein-based methods like Western blot, you can develop either polygonal or monoclonal antibody and target a particular protein, and from that you can do the blot and identify whether the virus is present or not. Some of the field side methods like lateral flow detic assay also has been developed. So like, for example, you can detect a single virus like white spot syndrome virus, single band if you find it is negative, and the 2 lines you find it is positive, or it has been also developed for multiple viruses like together white spot virus and yellow head virus. So depending on which virus is present, you can detect. And this will be very easy, particularly for the farmers, because you have to just add little bit of Korean your shrimp extract and then add some buffers or other things and all, develop, then you can know the result very quickly. So there are specific challenges in the diagnostic test. Suppose you are targeting, you saw a shrimp with white spots, you found that it's because of white spot. Further confirm, you will do the white spot virus, and you know that by PCR you got white spot virus, so you will be satisfied with that and keep quiet. But as you know, in the shrimps, there are mixed infections. So who knows, there will be other viruses also. So if you find a low virulence virus and you are satisfied, but simultaneously there is some other high virulent virus are there, then that time it will cause problem. You are not diagnosed it properly. Then when the new virus comes and all, standardizing the diagnostic protocol is an important aspect. If you are not initially you have to struggle a lot to standardize it, then only you'll be able to do the diagnosis. And particularly for new viruses to get positive controls. Say for example, the recent virus, decapodeuridovirus for DIV1. So many of the labs they are struggling to get the positive control. So to do any like test at least whether our system is having this virus or not. Then the cutoff, like which one you find that if this many copy numbers would be there, it will be important, and less than that it's not important. These things to determine, and in different countries case definition is different. Then next step is, suppose you are doing diagnosis, many private labs are there, many government laboratories are there. So whether all are doing the test correctly or not, how you do that. So there is one method called as ring test. So by which you can determine whether the test has been carried out properly or not. So what happens here, one lab serves as a nodal laboratory. So different grades of positivity, like control sample without any virus, one sample which has low copy numbers of virus. Suppose we may give it positive in the nested PCR only, but not direct PCR. Then medium level of virus or high level of virus, immediately in first step it will give. So different unknown samples is sent to participating laboratories. So they do the test, and samples are unknown, like given unknown code like A, B, C, D, or X, Y, Z something. You have to give the code numbers. They'll do the test and they'll send back the result. So based on that, you can decide whether they're efficient in doing the proper test or not. Then coming to the management practice of the viral diseases. So as you know, for the SIM viruses, so far there has been no treatment methods. So prevention is the best way, like how you'll keep away these viruses away from your culture system instead of finding some treatment methods. The first prevention method starts from the pond preparation. So many of the farmers, it has been seen because they are getting good production. So they do one culture practice, then immediately without giving any gap, they go for the next culture practice. They don't give any gap for pond preparation. They put the water, they keep the water inside the pond, give little bit of treatment, immediately they start the next culture practice, which is not good, particularly for white spot syndrome virus. So between the culture practices, you have to give the gap, take out the bad soil, treat it with limes, dry it for some times, then you should start the culture practice. So what we have seen, if you are not you're not giving the gap between the cultures, this white spot syndrome virus particularly, so they can be retain its infectivity even till 20 days. Even if you able to completely drain the pond, in that also, in some plankton or some other benthos, they can stay for about 21 days. And if the pond is not drainable, little bit of water is there, so in that condition, white spot syndrome virus can stay even up to 35 to 40 days. So you imagine, so you are thinking that you have done the treatment and you have get rid of the virus, but when you do the culture practice, again the infection starts and you lose your culture. So you should use the larvae from the genetically improved brood stocks, either it should be SPF, like specific pathogen free, or specific pathogen resistant, or specific pathogen tolerance, or SPT. So depending on like SPF stock, it is usually preferred because there the growth is not compromised. You just get rid of certain number of pathogens through screening and maintaining it for a certain time period. But if you consider SPR like resistance against a particular pathogen, say white spot syndrome virus, so there the genetic selection, you have to compromise either with the growth, generally with the growth. So generally this is not a preferred method, so it is best to avoid the disease conditions. So anyway, from these stocks you should be able to get the larvae. And another practice you should practice that to stock a large size of shrimp. Because if you see in general now, earlier in tiger shrimp, Penis monodon, they used to supply PL10 or PL15, even PL20. But nowadays in case of Vanamai, PL8 or 10, at that stage itself, they sell the larvae for the stocking. So in that stage, many of the immune systems are not yet clearly developed. Definitely they will be under severe stress. You are directly released into the pond. So it will be easy to get disease for this kind of larvae. But instead of that, if you can include a nursery phase in between your hatchery and culture, so that make them a little bigger size, then they will be able to be more resistant to pathogens. So they will be able to capable fighting with the pathogens, and there the survivability and getting disease survivability more, getting disease will be less. Then even if we say that is SPF stock or for double check, you should invariably do the testing for the larvae, at least for known virus. You know that WSSB, IHSNB, or EHP, or HPND, all these are important, and if it's there in the larvae, it can cause problem. So you can test larvae for all these pathogens. So once you find that these are negative, then you can go for stocking. Otherwise, you are carrying the pathogens with that. Then Next step, what you can do, suppose some of the regions there is already reported that some kind of disease is present. For example, at present, you know that DIV or decapitated virus is present in China. So you should avoid to bring any kinds of larvae, broodstocks, or live feed from those regions, because there's a high chance that you will also import the pathogens along with that. Then, after stocking, you should follow all the biosecurity measures, particularly the crab fencing, bird fencing. So these are, suppose somewhere mortality will be there, the bird can pick up one dead animal, and while flying, accidentally also it may fall in your pond, and that one shrimp is enough to finish all your ponds. So all these precautions you should be able to take, and you should have a reservoir system also invariably, so to treat the ponds, because nowadays particularly because so many culture practices going on, without any treatment, many farmers they release the water to the wild system. So that is also get contaminated. So directly if you take water from these systems, so there is every chance that those will carry virus, particularly white spot syndrome virus. It can associate with planktons and stay for a long period, or even for the zooplanktons and other things. So if you are taking direct water from the creek or sea, definitely the risk is very, very high. Another method is the zero water exchange or closed recirculatory system. So in that system, what you are doing, so if your reservoir system initially you can treat, so you are making sure that your water is good, and the same water you have to circulate. So you are totally cutting off your system from the outside environment. In that way, so you are making sure that you are not taking pathogen from outside. Then feed management. This is very, very important. How to manage in your system. So when some diseases comes, the shrimps becomes lethargic, they take less feed. So if that time you will go on adding feed without checking the check tray handle, so definitely it will deteriorate your environment, and shrimps will be under higher stress and it will cause problem. So at that particular time, you should be able to manage how much if is required. You should reduce the feed, particularly some other cases like AHP in the handle. Even if you stop total feeding for 1 or 2 days, that's considered as better. Coming to prophylaxis and therapy. As I told, therapy, there is no treatment for virus diseases. However, you can take many of the prophylaxis and other things are available, which you can take precautions. So among the physical methods, that is UV and gamma radiations. So through these radiations, in particular dose, you have to standardize, and it is able to kill the virus handle, particularly in hatchery condition handle. Many people they use this radiation handle to keep their water safe and then culture the larvae. Temperature also is an important factor for the multiplication of the virus. So by maintaining the temperature, you can maintain a good conditions, not allow the virus to multiply, and that's why chance of getting disease will be less. Particularly for white spot syndrome virus, you see some reports are there. So when you maintain a constant temperature of 32 degrees centigrade for 6-7 days, then you will be able to get a successful culture. pH, again it is best for laboratory condition only. I don't know how far it will be able to be applied in the culture system. Low pH like pH 3 handle, if you maintain, it will be able to kill the white spot syndrome virus. When the chemicals have been found to kill the virus, like ethanol, sodium hypochlorite, potassium permanganate, benzopyrene chloride, BKC, even sodium chloride also, the laboratory conditions they have tested. But for a disinfection process, you can take 2 of the chemicals like sodium hypochlorite and BKC. These 2 are important. So at least, suppose you are getting the stomach infection, so to disinfect your pond or in a reservoir system to disinfect the water and take up subsequently, you can use some of these chemicals. And many of the general and herbal-based immunostimulants are there. As you know, shrimp is having a non-specific immune system generally. So to stimulate the immune system, keep them healthy, so that they will be able to protect themselves against the pathogens, particularly glucans and the lipopolysaccharides, sodium alginate, chitosans, many of the nucleotides, peptidoglycans. All these have been used to stimulate the immune system of slim and get certain degree of protection. A number of medicinal plants are available, and those plant extracts have been also suitable to protect against pathogens. And many of the sweet seaweed extract, aquatic plants and particularly seaweeds, these serve as general stimulant, so they stimulate the immune system and they protect them. To some degree, they give protection. Probiotics is another important aspect, to maintain the diversity, and also they able to, through their external secretion, they can kill also many of the pathogens like Vibrio and all. They can control the secondary infection. That's why to when degree you can able to save your culture. Many of the bacillus species or lactobacillus species, they have been used as probiotics, and they have been found to be useful. Different biomolecules also extracted from species like actinomycetes or even crustaceans, seaweeds. So these biomolecules to the pure molecule level, they have been extracted and then found to be suitable for against many of the viruses, particularly white spot syndrome virus. Vaccines also have been developed, like taking particular proteins. You can develop monovalent vaccines or polyvalent vaccines. Particularly, many of the vaccines are available against the BP28 gene of white spot syndrome virus, and then based on laboratory conditions, it has been found to be effective. In inactivated virus also, it has given certain degree of protections, plus DNA vaccines. However, all these things, they have been carried out only in laboratory-based conditions through injection methods. But here, it's required to develop a field delivery technology to take it to field. Similarly, many of the antibody-based approaches, directly that antibody developed against white spot syndrome and that given to slim, it is able to neutralize the viruses, or egg yolk, which has been immunized with white spot syndrome virus. Those immunoglobulins, they have been found to be also neutralize the virus. Nanoparticles handle like chitosans or polyanidated nanoparticles, they can encapsulate the vaccines or the fluorescent RNA. This can be used to apply in the field. RNA interference also, RNA interference mechanism is available in shrimp. It has been established, and many of the reports you can see, whether it's the fluorescent RNA or siRNA or microRNA. So all this, to some extent, they give protections. Laboratory conditions, many reports show that they give 100% protection against the virus. But again, this also not been possible to take into the field because the field delivery mechanism is not there. So the shrimps want a definite quantity of double-stranded RNA to provide protections. And recently, CRISPR-Cas-based methods have been developed, and many is still under trial. Many people are trying how to develop a method based on this technology to take it into the field, provide protection against virus. So to conclude, now aquaculture is very important. Capture fish is coming down. So the aquaculture is the only future to supply the animal protein to people. And so aquaculture will continue. And also as the human population will go, then people will try to get more and more production from the limited area with the higher surface density and all. So disease will invariably a part of this system. So with the intensification, and based on that, definitely different new pathogens will come, and many carrier animals will be also there. So you have to take care by developing improved diagnostic methods, your surveillance method, disease monitoring protocols, and nowadays, like as you know, nucleic acid-based assays, they have been found very sensitive and specific. Within a short period of time, you can do, and even through multiplex assay at a time, large number of pathogens also you can able to detect. So with that, I conclude my talk. Thank you very much. If you have any questions, I'll be able to.
Thank you, doctor. I'm from Hyderabad, doctor.
Yeah.
And you know, now Telangana is emerging in aquaculture,
Yes.
inland fish farming. So, interfaces have.
Yeah.
So we have to do this shrimp culture in tanks that is above ground. So I would like to know what precautions we have to take in such
Water you are using, you should have a reservoir system. Though the freshwater system are much better, because many of the pathogens you won't get. But still, once you go on culture, doing the culture practice in a particular area, so like for example, I'll say when I came for a few years, there is no problem at all. But subsequently, the problems again get started like that of modern culture. So the citizens get adapted to the new environment. Then they cause problems. So even if you are doing that system, you should have a reservoir system. So make sure that your water is free from pathogens. Then you should make a good diversity, like a natural system. You should create a system so that is suitable for the animals. Then go for culture practice. Then there is less.
suppose we are using borewell water, doctor, because reservoirs are also not many over here. So we are, if you take monthly, using borewell, and the borewell water is tested for acidity, alkalinity, and everything. So here, what we can do is we can culture in this. What precautions do we have to take? Same thing, like if
water means there will be some kinds of mineral deficiency. So whatever you get from sea water, all the salts not available in the bore water. So streams need also particular. So externally some particular important like calcium, magnesium, other things, that balance you have to maintain. Plus you can go for culture payment, that's no problem. But again, so stock intensity you have to standardize how best you can go. So that will initially it will take you some 1 or 2 times to standardize how much stock intensity or little minerals are available in that bore water that will be utilized by the majority of the streams. So like if you know that the running water is the intermediate occurs mainly because of the mineral deficiency and other toxic gases. So these things initially you have to standardize and
where can I get a literature on what the mineral should be added in such cases in bioprocess?
I think in the second part talk today there will be another talk by Dr. Pani from our institute who will talk on bioflow. So whatever your doubt regarding bioflow, you can ask him.
One more question, sir.
Yes.
When I'm talking, I do not find the PL. What is the best size PL for me to transport? I mean, I have to get myself from
Unfortunately, you don't get a choice. The hatchery people, they sell in that particular PL8 or PL10. So you can take and do a nursery phase and you can do. Or like this for known pathogens.
non-pathogens.
Yes. Because many of the hatcheries, what they do, illegally they do the period bootstraps and they use it instead of the SPK bootstraps. So in that case, you get getting disease very high, particularly nowadays at EHP and IHHNB, all these things you get. So better to test and then take. Okay. Welcome.
Hello, doctor.
Yes, sir.
Sir, we have known that the vaccines and antibody capsules are discovered
in India, for India to get the vaccines, as the farmers are now very much interested to apply the medicines or the vaccines
No
for virus resistance.
either in India or any part of the world. Whatever has been done is only on an experimental basis, laboratory. As I told, though it is been established in the laboratory conditions, taking this that has not been standardized. So commercially, it is not at all available.
Okay, sir. Thank you, sir. Thank you, sir. If there is no more question, I thank Dr. Gupta sir for his excellent presentation as well as the experiences. I thank you, sir, for your excellent
Thank you very much, sir, for the opportunity.
Thank you. Now I request Dr. Vidya, you are there. Dr. Vidya Rajendran, you are there. Yeah. Kindly make experience of Steve and their management. He is an excellent worker, doing mainly on the bacterial disease, and he's scientist at ICFRI Chennai, and his main work is on the microbial interactions in ecosystems, disease resistance prophylactic using bacteriophages and probiotics, etc. So I request Dr. Vidya to continue. Please go to the present now. Go to present now, then share full screen. Madam, go to present now. Is it audible? Not audible? Hello?
Hello, sir.
Yeah. Please,
Share my presentation.
yeah, go to present now, share the full screen, then go to the folder and open the file.
Is it visible now?
No, it has not yet.
Hello, sir.
Yeah, it is coming.
Yes, sir. Yes, sir. Thank you so much, sir.
Can we
Good morning to all. Yes, sir. Can I start? I'm Dr. Vidya, according to Environment Division, Central Institute of Brackish Water Aquaculture, would like to present here about the importance of water quality in shrimp farming, the bacterial diseases usually found in our shrimp farms. So, as we know from the last presentation, you know that but high income generating one. So now around the world.
Doctor, your voice is breaking, doctor.
Is it audible?
Good morning, doctor. Your voice is breaking.
It is breaking in between.
Wi-Fi. So can I little bit slow down while speaking so that it will make up? Little bit it will make up. Hello?
Your audio is breaking, doctor.
How is right now, ma'am?
Yeah. In the beginning it is fine, but when you continue talking, it is breaking.
Because the speed of the way which I speak, maybe. Is it okay right now?
Yes, doctor.
Yeah, I think I have to little bit slow down, then it will be here to everybody. Okay. Then I'll. Okay. We just continue from.
Continue.
The mushroom farming is very risky, but it gives high income.
Please continue. Your voice is not okay.
Yeah, there's so many risk factors associated with mushroom farming. Mainly, we can correlate to water quality. So far, it has come to a commercial level. So the risk comes from the seed quality and the water quality. Once the water quality is weak, the pathogen interaction to the host, that is shrimp here, can be we can control to a
It's not audible.
level.
Madam, it is not coming properly. Only few words are coming. Could you hear me? Hello?
Shrimp pond ecosystem. How
Madam, your voice is not audible. Could you hear me? Hello? ये ऑनलाइन का ही चक्कर है. Madam, your voice is not coming. Madam, can you close your presentation and then again restart? Madam, no. तो मोबाइल से प्रेजेंटेशन कर रहे हैं. minutes, Madam will be trying from another system. She is switching to another system. Please give her some time, 2 seconds. Today, after Dr. Vidya's presentation, we have Dr. Panilai's presentation. Then we will go to presentation by Dr. P.K. Sahu from CIFRI on albulosis. Then in the second half, possibly we have to go for the feed and feeding management class. Then we will have valedictory. Am I audible to everybody?
Yes, doctor.
Yeah. Please,
Yes,
Madam is switching to another system. Please give some time.
Okay.
How you are feeling about the training? Is it going on? Okay or not okay? अच्छा लग रहा है या नहीं?
अच्छा लग रहा है डॉक्टर.
It's really good, sir. We need this type of training. Thank you, sir.
Dr. Mana, you were telling about lecture on by somebody on RAS system because
RAS and Dr. Rajesh Kumar from CIFRI Bhubaneswar, he is the man working on RAS. But he can work. I had a long talk with him yesterday. Okay. So, but he cannot have time today. That became a problem. And nobody else is working on RAS.
so when can we have it in the future?
Hello? Okay. I am trying. हाँ सर मैं सर अभी आप वो थोड़ा मैडम का कनेक्शन का थोड़ा प्रॉब्लम हो रहा है, आप अभी देख पाएंगे? हाँ प्लीज सर ओके now instead of Dr. Vidya Rajan presentation, she is facing some problem in connection. We will have presentation by Dr. Panilai on biofloc. He is a man of biofloc, widely known in India. He started biofloc in India with Binami, and later on the biofloc has been extended to TDS also. Some maybe from the other high pieces. But he basically started and still expert in this field. So we could hook him and he was glad to have a presentation. And in biofloc system, diseases are not any specific disease. It is the species, part of our culture, it is the disease of that species. But water quality and day-to-day management is very critical in the biofloc. So Dr. Panilai will be explaining that how to go for the biofloc, how to manage the water quality, how to manage the nitrogen phosphorus balance, etc. So please wait for a few seconds. He will be soon coming. Please wait. Just give some time to make him present.
Sir, today today Dr. Hassan will give the presentation.
Yeah. In the second half, we can go. The online problem, this is the problem. We are totally dependent on the connectivity. आज जो ऑनलाइन में है, those who are present here, today we will give feedback form. In that feedback form, please mention your email ID. We will send the participation certificate in that email ID.
Yes, Doctor. Yes, we'll do that.
We will be sending the participation certificate in that email ID.
Please repeat.
We will be sending the feedback form. In that feedback form, mention your email ID clearly.
Okay, sir.
So, Dr. Manna,
Yeah, audible.
yeah. So, happy morning to all the participants, respected Director CIFRI, Dr. Manna, and all my seniors. I came a little before. I think the earlier audience had some problem, internet problem. And so this is a topic on health management in biofloc system. Actually, these are all like two paradox things. When you think of biofloc, and many things are going on. The rate of growth, if you consider, just in 1.5 years, there could be 2 biofloc units. Now there are 700 biofloc units. You can imagine the kind of growth, attention from the real scientific world, and it is not really getting that way. So, without proper training, without proper this one, whoever start biofloc having a tank, I think they are just next to your charging. I know 3,000 to 5,000 rupees just to show the tank and some aeration and some air bubbles, and it is all that way it was going. So now only things are getting better, and people, there is every aquaculture new technology, when it comes, it needs a course correction, and in that process properly it will go. We know that. And now coming to all the time when we talk of the heterotrophic bacteria, these heterotrophic bacteria, these are all like this differ from way. In conventional system, we always think of mostly plankton based system. There the plankton is the hero, and problem is that plankton is very good for water quality and all this thing. Only thing, beyond a certain proportion, it cannot feed any input you are giving
giving in the system, it ultimately boils down to 6 feet. Whatever protein is there, 70% goes to the environment, and out of that 17%, almost like 16% is nitrogen. And nitrogen, all the toxic metabolites, whatever we can think of in the water, there it is handicapped. When the plankton can only absorb 6%, 6 to 10% of the whatever nitrogenous metabolite. 40 years of research at research level, and also then finally it goes to the commercial in France and in Texas, in US and all. So this kind of system involves bacteria who are good in eating these or scrapping all these are called heterotrophic bacteria. They have the tremendous capacity to absorb all the nitrogenous metabolites, whatever is there in the water. Any carbohydrate you take, for example, carbon is there nearly 40 to 50% carbon will be any white flour and even the molasses, jaggery, everything is a carbon, including sugar, everything is a carbon source, carbohydrate source. So that if you give in excess, then this will be eating away all the nitrogenous metabolites. So what it gives is the in situ bioremediation. It means whatever nitrogenous metabolites because of their thing is taken by these bacteria, and these bacteria are so fast in their growth rate. They grow very fast. After an hour to 1 hour, they will be next 1 lakh, become 2 lakh and 2 lakh. That's a geometric proportion. Pardon? Dr. Pani, yeah, have you your presentation? Yeah, I am yet to have you loaded? Yeah. Just introductory this system. So let me also do this share this screen. Is it visible, doctor? Yeah, yes, it is coming. Okay. So, so this is what we are talking now. This now from the plankton, we are in a biofloc or in a heterotrophic bacterial world. These bacteria are very good beneficial bacteria, not only to finish your whatever waste or whatever these nitrogenous metabolites are there. It can also give give the system a very stable environment, because plankton is for its growth, for its everything, it depends on this solar light, and it will definitely early morning hours in the system they will absorb all through the night. They will absorb all the oxygen, but without production of this one. So early morning pH will be low, the temperature research is low, and then the other things like the dissolved oxygen also is low early morning. But as the day progresses, the environment changes in a plankton-based system. But in a bacterial system like this, there is no change. It gives a very good stability. So now coming to this is a very stable environment from where the waste is converted to bacterial protein, and the bacterial protein could give a very good this one. They are also a very good protein source for the reared animal, and also simultaneously, because these bacteria are beneficial bacteria, they are kind of probiotic bacteria, so they are natural probiotic and so immune. So you don't need, this is a probiotic, basically a probiotic system where antibiotics, we always have a zero tolerance for any kind of antibiotics. So when you talk of health management, first thing comes, this is a very alternate health management system where we don't allow, or we should not allow antibiotics in a system, in this biofloc system. The moment you apply antibiotics in the biofloc tank, the biofloc itself, whatever are there, they will also die, and it will be, the problem will be compounded. So then, this is a natural productive system. Only thing, we have to see the better management practices. What better management practices possible to create still in this biofloc environment, how to maintain a proper health and a proper environment by following the better management practices. So when we talk of disease, that is the biggest challenge in the goal survey. You see, whatever they do the survey, disease rank the first concern of any farmer or anybody is disease. Coming to India, now we are the best country to producing the shrimp, almost like 8 lakh metric ton, and equally the fish and shrimp production. If you see the way it is growing, I think the fastest food producing sector. And so here, what is just our take is, those who are familiar with shrimp, we have certain diseases like white spot disease, the ESP and IHMV. These are all some other AINP. These are all, every 6 months there is some emerging virus or bacteria. It comes and it destabilizes. Even now the microsporidium and EHP, which causes the white fecal and other syndrome. So that also is there. So we just recently, we have one publication where we have just tried to figure out what are the kind of loss India is facing from the 40,000 crore. Of course, we are running from the export of this shrimp. But if you see, because of the disease, at least that the probability of infectious disease occurrence is 49%. So that is the loss we are having because of this kind of, only in one sector, that is the shrimp aquaculture sector. I am telling each disease wise what is the probability, and this one is given here. So what are the way to do alternate health management systems we tried to promote earlier? One is organic farming system. We have demonstrated every sphere, but here it is not taken up that much step, only because the production limit. Here, in organic concept, we think of the health of the animal or the well-being of the animal. So there, we have to reduce the stocking density and the production limit, and that is why, even though you are getting a higher premium price, but then you can see the natural production and the hormone-free production and organic is the highest in the pyramid. You can get a problem, but then now we are attempting to define this biofloc system as a natural production system or natural as organic farming system, because as long as we don't use this kind of antibiotics and the banned chemical system, so that is how one of the important thing. And the probiotics, what is its advantage, and when you use antibiotics, it the animal loses its immunity but probiotics always enhances immunity, at least, though the performance wise both work similarly, but in the production system, when the challenging situation comes, the probiotic based animal, they perform much better. So these are the earlier to this biofloc thing, the concept how our Indian farmers are attached to. And now, with probiotics and the fermentation and prebiotic, another feed component which promotes the probiotic bacterial growth, the symbiotic concept. So what we are seeing is all these take it for example, bibliosis or early mortality syndrome or white fecal or WSSP. When we do use this kind of symbiotic system or fermented product to use that bacterial or yeast fermented, which all the farmers, organic juices, everybody know how to use that one. Indian farmer for last 20-25 years they are doing. So this is all the just before that biofloc. These are the systems which are prevalent, which says that we can better approach the health challenges following this kind of system. So now coming to the biofloc system, as I told you, this is a concept of naturally this bacterial protein system. These bioflocs are very rich in protein, and also this is a high level of amino acids and the polyunsaturated fatty acid and bioavailable organic mineral, vitamin, enzyme, everything is there in the bioflocs. And so it means to say that you do not worry if you are having a proper biofloc system. You need not worry for its neither for its disease problem or for some kind of using many other vitamins, many other things. I think this is naturally that kind of system. This is and what are the advantages? We have already discussed. This biofloc system reduces the fluctuation. It doubles the protein utilization. I told the feed protein first time it is utilized, again it becomes bacteria protein, again it is used. So that's a very pure form of protein, and which gives also because of the natural probiotic effect. So these are all the advantages, but equally it is a very technically demanding technology, and also this is high aeration because the bacterial biomass itself is almost 3 to 4 times of whatever your animal biomass. So you need to give more of bacteria and more of oxygen in this system. So we have we are demonstrating this nursery and also the growth system where we have seen the survival in case of shrimp nursery, almost like 94% survival. And also in a growth system, you can find in this thing all before 2013.
Sir, is it possible to enlarge the screen?
Yes, I think is it okay now? Is it okay?
I mean, it can be whether it can be full screen or slightly bigger screen.
Now here mine is full screen. I am showing full screen.
Now can you go to the listing mode, tile mode?
Go to the
Tile mode. PPT presentation. I mean the slide presentation, slide preparation mode. Tile mode.
Slide mode, sir.
Yeah, slide. So only I am putting.
No. Left one. Left, most left. Yeah.
Yeah. Now most left it is there already.
Then go to the slide presentation mode. Yeah.
Slide so is now it is big screen here.
No, it is not coming here. I mean, actually, it is not readable. Actually, if you are try to read it, it is not. This slide is readable, but the other slides are not readable.
Now, okay. Some slides are not readable.
Yeah.
It is not showing now. It is showing this one. Okay. So I'll just I'll call.
Please continue.
Okay. So should I once again share?
No, continue.
Okay. So this is one of the production system where we tried to in 2013-14. Hello. It's the slide very size it's only a small slide. So this is here one comparison we can show that biofloc system, how from the conventional system we did with 110 days of culture that time in 2013-14 when we did, and now it is much advanced way. So there we get a average body weight of the shrimp is becoming 36 to 40 gram 110 days following the nursery, whereas in the conventional system it is 25 to 30 grams. So there is an enhancement of almost like 27%, and similarly the survival improves like almost 20%, and productivity, if you see in the conventional system when it is 2.5 to 3 kg, they are not able to see the big screen. Even important. No, it's not coming. Doctor, can you see bigger screen?
Yes, one is going on.
So, and the productivity, if you see that way, there is an enhancement of productivity in the biofloc system around 52%. It's I'm telling this is one of our study, and which is also okay. We are not able to. It's not able to fix. So here we are actually. I called my computer expert, and he is telling from here we are already in the higher slide sharing mode.
Okay.
In the bigger screen, but I think why it is not going to see that. Okay, so then this is the FCR is also equally reduced in the biofloc system because the feed is giving a very good protein, so FCR is reduced. After that, we have been doing a lot of study, and we know the carbohydrate source. Many part of India we don't get the proper carbohydrate, and with this particular carbohydrate, which is important for from the health management point of view, that is the digestibility of the carbohydrate, or the how fast it can be utilized by your heterotrophic bacteria, that varies. And accordingly, some carbohydrate are complex carbohydrate, they will not be utilized very fast, so it needs fermentation much before you put in the pond or in the tank. You need to do the proper fermentation with proper kind of bacteria, and there are some kind of bacteria which produces more of amylase or protease or glutinase or lipase. So those are the way by which this carbohydrates, whether you are using jaggery or sugar or any complex carbohydrate, accordingly it will give the kind of benefit, and one of the major benefit is the kind of bacterial maintenance of the bacteria and how they are giving the immunity of this one. We have also had some good studies in this aspect. Then this is another some few experience we have in Indian farming sector with whom we are collaborating, and this is a particular example of the farming where you can find the production system where it is up to 47 ton per hectare. That much we have achieved in case of shrimp, and also the FCR is beneficial, and this one compared to the conventional system, which is much less. So now the question comes when this system is so advantageous, and this one what goes wrong? And in a way, if you see, you take it for fish or shrimp. This is the classical diagram. I think many of this one would have shown you. So here, this system is more like health. It can be maintained here easily because this is a biofloc. It's a small system, and this is a biosecured system. Always we tell it's a biosecured. Is minimal because since it is a smaller unit, and you are also, it is a high production system, you are supposed to maintain all the biosecurity and all this thing. And the animal you are taking, that is another input animal you are taking, the health status of the animal, that is most important. In case of shrimp, we check for all the viruses, whatever possible now, WSSV, AINP, or this IHHNB, and nowadays the ESP1 microsporidian again added to that one. So you must check all these virus or pathogens before putting in the system, and the environment also equally important. The water which you are taking, my friends, those who are in the fresh water, we know they can manage even only this disinfection the way they think is only add some salt. They add some 1 kg salt per ton of water, and with that they think it is disinfected. But when we think of
Doctor, sorry to interrupt, Doctor.
Yeah.
Are the slides moving, Doctor? We are on 10th slide.
Now, can you see the other slides?
No, I think you are showing 10th slide. I think the slides are not moving, Doctor.
Okay, so Just let me see that.
Sorry to interrupt. It will be better if you present the slide.
Can you see it now?
Now we are in the 11th slide, right?
Okay, but whether it is moving, can you see?
Now it is moving down. Now it is.
Okay. Okay, thank you. And is there any improvement in the size of the screen or this one?
It is better, doctor. Now it is better.
So this is what we were trying to tell the productivity, and this is when we think of this production system. Here we are thinking how the interaction between the pathogen, the animal, and the environment, which dictate the terms. But it is all the, if you see world over, they say this biofloc system is the panacea for all kinds of diseases. You don't encounter any diseases, and when we conditions, we find the biofloc, it's not easy for any other virus bacteria to ingress and to cause any big mortality of this one. But in the field, it is not translated that way, because rather, and because when the pathogen somehow get entry, this media and all this kind of carbohydrate supply, that also is a conducive, gives a conducive environment for pathogen also. Hello. Hello. Okay, so this is what this kind of diseases it can in case of extreme, at least the WSSV and HHNV, as you have studied, and in case of peace, we know like the bacteria, the fungal disease, and this one is very common, and this is all our own. Some places also we see the operculum divatum. This is a species who is generally is a brachystoter milkfish. We say that one of the farmers was doing, and in high density, this kind of, when there is a deficiency of vitamin, it causes like this kind of disease. So many kind of diseases are we come across. But what are we just because time also is short, I'll just touch upon the what are things from health, the better health or production point of view, which are the points which are important, and in a way, as Dr. Manna also told, to treat here, because the system itself, you have to, the prevention is the best thing here. Prevention is better than cure, and we have to, like, as I know, like here is a system where the slide is moving now. Hello?
Doctor, you are in the 11th slide.
No,
sir. It is
It's not moving, sir, again.
it started moving.
No, doctor.
no, sir.
So this is actually.
Can you do it manually, doctor? Manually go to each slide.
Manually only I am going, but I think it was when it was whether your screen is bigger, you can see here bigger screen.
No, sir. It is still in last.
You have to present your screen, doctor.
Screen is not coming.
Now the screen is coming.
Yeah, it is, sir.
Yes, doctor.
And you can see
Okay, it's 17.
the slide is moving.
If you are doing manually, it is moving, doctor. Otherwise, it is not moving. We were only in the 11th slide.
Okay. So 11th slide onward, I'll just proceed. But when I'm with full screen, I think there is a problem. It's not moving. But when I'm small screen, I think maybe it is coming. Okay, so this we are talking of the classical diagram of the fish pathogen and the environment interaction, and this is what the diseases which are very common in case of shrimp aquaculture, and we just talked, and in case of fish, this kind of disease in the biofloc system, we are coming across this kind of fin tail rot, the dropsy, the other things are we are coming across, especially in the biofloc system, and in the biofloc system also the gill porcular divertide and all these things, because here, if you compare to a pond system, in a pond system, pond culture, we just maintain, we take for example any fish, it is only 0.5 to 1 kg per cubic meter of water we talk of. But here we are talking of 20 to 30 kg of, let's say, magut or singi or anything, or tilapia, 40 to 50 kg of tilapia, or pangas, maybe 60 to 90 kg of pangas. We are talking of the density or the biomass is at least 30, 40, 50 times more. So very much likely that there is a possible problem. It could appear health condition could be challenging. So we have to take all these steps for better management practices. There it goes, some certain sites are very difficult, and the groundwater, especially in India, they have a problem of very high alkalinity and very high ammonia nitrite itself in the water itself. So unless it is amended properly, you cannot take it for your culture. If you are taking also, there could be some problem. So water quality profiling is very important before you proceed for any biofloc kind of system. Then this system design, here the bacteria grow very fast. As I told, every 1 hour the bacteria doubles, and the production of this one, and equally they will be dying, and there will be some sludge formation. That sludge settles in the bottom, and it should be promptly removed from the bottom. As and when it is formed every day by routine, it should be removed, because if it is that is why all this system design is important. There should be a central drainage kind of system which will give rise to the exclusion of all these sludge management possible properly. Then species specific SOP, that is also the which species you are talking. Suppose immediate now farmers are very much telling, if you are doing pangas, initially when you stock, you don't need that much of aeration because it works in a negative way. And the quarantine facility we talk, some species definitely they during the transportation and all, they carry a lot of these parasites and this one. You need a hospital facility, a hospital tank, they say popularly, or a quarantine facility in your small tank where you can treat or keep the fish for observation for 2-3 days before you are going for really going for stocking the biofloc pond, because stocking this unhealthy in this biofloc system. Then the biofloc generation, that is really a very good aspect which should be given more emphasis. And quality seed, then water management and health management, feed and flock management, sludge management, then disease. Invariably, in spite of doing all this, you could also end up in getting some diseases. That time, how to address those diseases, how to take out the fish and treat if it is absolutely required, or how you can do in the biofloc volume. And because initially you have the problem of generation of biofloc. After seed and your nitrogen is more, as you add this carbon, biofloc and enormous biofloc develops, and that only brings all kind of problem. So you need to check the biofloc, that is what, and the harvest and post-harvest and effluent treatment. So this is all like in short this selection of the site, as I told, the water quality and amendment, what is required for the ammonia, alkalinity or iron or other pollutant, then the selection of these species. Here, more than your this one, the availability of the healthy animal, healthy seed, that is, and these of course here one of the major thing is economics. You have to see your market, which now like the anabas, we had a very good market, and now with the biofloc developing everywhere, the market they lose, and this one you have to see which best species is value you can get for the money, and then the even for the particular species, doing it in phases, like nursery introducing in a phase wise harvesting. If you have couple of 50 tanks or 30-40 tanks, you should not at a time stock and at a time. Instead, you can have a crop calendar, and you can do that, and that will help you also to break the chain of any virus or bacteria for that matter. And the design and infrastructure support, as we have discussed, the proper central drainage system and adequate aeration and power backup, all these things are important. As I've shown these, some people they combine this biofloc with periphyton. Then periphyton can be or another substance like similar to biofloc, but they need some kind of substrate where they settle down and the animal can sit over them and eat. So this is what the periphyton nets can be plugged in the system, and that can give a symbiotic system of biofloc and periphyton. And you can see in that figure how this the water can be moved in a centripetal manner, and then the central drainage system how it this. Then there are many kind of different kind of tanks, but the whole philosophy here is how to take out the slogs or the waste generated on the daily on the routine basis, and that is what we are looking. And then it could be round, it could be elliptical in shape. We tell also resuate type of biofloc. Here we have some design where the water also flows from bottom aeration and also simultaneously. So aeration is one of the most important thing here. Many places it fails because you are not only giving for the animal, the bacteria also need a lot of aeration. So that is one of the things. Once your system is ready, then the water you are taking, at some places only filtration. Filtration is a very important aspect to take the water to your system. It should be filtered like for the animals like shrimp and all, which are very much exposed to different kind of virus bacteria. So the any carrier should be, you have a series of filtration net before taking the water into the system. Then in case of freshwater, we know very with salt treatment or potassium permanganate we can manage, or maybe maximum with bleaching to treat. But with the different kind of diseases coming now, suppose as we are talking of the EHP and this one where pH fluctuation, pH oscillation is very important. There we try to disinfect the water with this kind of how to increase the pH with calcium oxide, with sodium hydroxide. We try to increase the pH, and then otherwise with hydrochloric, with chlorine, liquid chlorine, and this one. And potassium permanganate is known to be very good as a treatment for this. So many areas where it is affected with a kind of bacteria or virus or microsporidium, accordingly you have to decide your disinfection protocol. Then the biofloc generation, of course, many things are there, but basically 3 ways we can develop the biofloc, which is the one is you have the old biofloc, you just need to a proper biofloc, you need to inoculate the same biofloc and maintain the CN ratio, it will be developing nicely. You have to start with a proper inoculum, with proper fermented this FCO, what we tell. This is with that also you can start, and for that the probiotic you are using, that is most important, how good they are, what is the number they have, and how much billion cells they have per gram. That is most important to select that one also. If you are not using in a classical biofloc system, no probiotic is required, because here is a system where we encourage diversity of microbes. Once you are giving customized probiotic, then you are challenging the diversity of the system. So diversity of the is a strength. When you talk to people like Europe and all, they don't know what is that good probiotic or good heterotrophic bacteria. But generally, our farmers, they want to kickstart the this system very fast. They want to generate the biofloc. That time it is required. And another system, you go very slow. First with algal culture you develop, or the plankton-based system. Then slowly when your feed quantity or the ammonia generated is more, slowly you try to put some carbohydrate, and you can also also develop some kind of very good biofloc system it can develop. Once the biofloc is developed, then how to go about the how much addition of the carbohydrate, and very simply that 1 ppm of ton total ammonic nitrogen need almost 20 ppm of this carbohydrate. So for every gram of nitrogen this total ammonic nitrogen you have 20 gram of you have to add the carbohydrate for that matter, and you can calculate how much ppm of ton is there, and there are, but then your feed also, feed is the protein of the feed is more, then your CN ratio of what you are getting from the feed is also getting is less, so you have to add more of carbon. So it is always useful if you are using protein with less protein feed, if you are using, that is always useful. Now coming to the health seed, in case of shrimp we talk of, and quality seed means Vibrio count should be less. Here we talk less than 10 to the power 3 green colony per gram of PL, and then the developed yield, at least we tell PL10 you should take for stocking, not before that, because before that the yields and all are not properly, because nowadays to say, because people even PL6 before that also people started picking, you should always see PL9 to 10 for Venamine seed. Then you must do the salinity stress test and other this one like the gross examination. That should not be when you look under microscope the seed the PL make a current in a small pot. This should go against the current flow, and then they should then when you do the stress test, like reduce the salinity by 50%, you find that 90% of the animals still survive after a few hours. So these are the gross examination you can do, and this is how the under microscope you can see how the golden yellow head pancreas says that these seeds are very healthy. Likewise also the muscle gut ratio, the muscle should be good, and in that case the muscle gut ratio it should be good. And so these are the way you can select good seed. The so is that the animal which you are taking, like many people from our place, from West Bengal is the hub of all the breeding centers and all. When they carry to long distance, there is a stress and this one. So there should be a proper protocol they should follow, and immediately they put in potassium permanganate high dose, and there itself before stocking the animal they start using the animal. So those are all important to see how best they can transport also without much stress. Then water quality and flock management, that is one of the important aspects by which you can reduce the TAN level and the alkalinity you can maintain. Here, once the biofloc develop, the alkalinity drops very fast. 100, it will reduce from 150 to 100 to 80. So you should always add this any lime material to make up the alkalinity. That is one of the important things. And this nitrite and TAN should be at, and then the biofloc volume for seed with that, and after that only 10 to 15 should be there. But for fish it can go little higher, 25, 30, 40, but not beyond that, because the total suspended solid, if it goes to more than 300 ppm or 400 ppm, it is very dangerous, because the gills will clog and the animal will be stressed to have the problem. Then the slough management, here we already discussed sloughs, how it is dangerous and how it should be rooted. Here almost sloughs is accumulated over the period for 1 kg production of this one. So it should be properly disposed, and otherwise the sloughs itself will bring down all the protozoans and all these things into the system. Then the growth and health monitoring, how to do that. Disease occurrence also, most of the time what we try to add more feed, because in such of this is because we overestimate also the production expected. It is not, there is a carrying capacity, it cannot handle beyond that. So by regular sampling you can know also whether it is properly going. And then like I told in gist, what you want to tell is the water quality improved, water quality should be maintained, and whenever there is a problem, You have to find out the root cause of that. Instead of putting immediately some what I find is many farmers, they are excited to put this enrofloxacin or the oxytetracycline or tetracycline, all those things. They are not a solution. For at least in a biofloc system, please do not add these antibiotics in the system, because the biofloc will collapse and the system will be unstable. You can never bring back this one. At least if you want, or if it is unavoidable, you take it out and put in a other tank and treat it and this one. But it is always best to avoid this kind of medicines always. And then by supplementation of vitamin and mineral, and this was also, that is of course like vitamin C, that's an anti-stressor. You can always add vitamin C or vitamin B complex. That is what people are doing also to make the system or the animal more healthier. And the feed also should be very regulated feed, not underfeeding, neither overfeeding. And whenever there is ammonia problem, you reduce. First thing you must do is you must reduce your feed. Don't feed, and even fasting is much better. Like us also, fasting is a good therapeutic measures like that for shrimp, for fish. You can do them fasting instead of overfeeding them. Then when there is a crash of this one, sometime because of the natural sunlight, plankton and all these things, the crashes, and that time also feed immediately should be reduced. And there is a problem of DO, feed should be reduced. Temperature fluctuation, feed should be like temperature reduced by 3 degree, 4 degree during the cloudy year, then immediately 30 to 40% feed should be reduced. And like that, molting time also for shrimp feed is reduced. So what is our main is that whatever the stress causing factor in a biofloc system, it could be because of the total ammonium nitrogen, nitrite, or other contaminant or sulfide. Because if this clause is not removed, they will form into some kind of sulfide. So it should be immediately addressed. And prevention, as I told, is better. And then we can go for how to reduce the stress. And whenever you are using the healthcare product, also you should use most judiciously. You should properly understand what for the product, what the problem you are having at hand, and just because somebody is telling you should know from a certificate. But in a biofloc system, as such, the animal is more healthy. From this graph, we can know that when biofloc system, it is definitely the mortality in the control system, we can see when it is 90% in the biofloc grown animal, when it is challenged with pathogenic Vibrio parahemolyticals, we have seen it is only 40%. The mortality is 40%. So they are naturally healthy, it means. Then the addressing the loss of immunity. So this is what optimize the bottom management. Like here, this loss management is important. Because of that, this one, then oxygen level should be always more than 5, and take care of the spike of total ammonium nitrogen or nitrate by CN ratio manipulation, and stabilize the pH. As I told, the alkalinity should be also addressed and stabilized. These are the animal got when it is reared in biofloc. It means food is available 24/7. Here, the bacterial protein food is available. You need not worry for giving excess feed or this one. Whenever there is a slight hint that you have a problem, you reduce the feed 50-70%. That is the best approach here. And there are so many success stories, though I know many farmers are having tough time. There are so many. Here is something like in Paku, and in case of Scampi, we have seen the survival is 88% and 97%. And there are so many success stories. That's why only people are motivated, technically more demanding system. This is one of the biofloc pond where you can see the kind of aeration it is given. There is in the center, there is a water wave creator. There is all through, there is almost like 50 to 80 HP capacity of aeration in a 1 hectare pond. That must also be required to get a proper biofloc. And there are so many things we can.
Sir, may I have the previous slide? May I have the previous slide?
Yes, this one.
No, previous to this.
Yes.
Yeah. Okay, sir. Thank you, sir.
Yeah, thank you. So this is what is then every aspect. If you see rain, during the rain is also it is important, as I told, during the winter. Winter, as the temperature drop 1 degree, 2 degree, 5 degree, your feed at 1 degree reduction, feed consumption will reduce by 10%. Like that, when it reduces by 8 to 9 degree, and from the optimum of 26 to 28 degree, definitely the animal will almost stop feeding. So you have to observe that and don't feed that time. Like that, when rainfall is there, when the alkalinity drops because of the rainfall, acidity improves, and there also you need to, and during the rain, especially for shrimp and all, even for fish, the rainwater, if it is falling on the tank, you must be very careful about the minerals supplement will help you in a big way. Then the fermentation product you can apply more often and reduce this whenever there is a continuing rain, you have to stop feeding or reduce feeding by 50% at least. So these are, I'm telling of many aspects could be linked finally, which will give you a healthy harvest and healthy animals to harvest. And the climate change also, you know that this is a climate smart aquaculture system. We talk this biofloc system with this minimum water requirement and everything. It is a climate smart aquaculture system, and it should be also approached from the at the farm level and the system level, and to get this one. Then another important thing, when we have this biofloc slough removed from this one, instead of that, if during the biofloc culture itself, you can take out the biofloc generated in the water, and we have developed some kind of harvester, and that is you can use this biofloc by drying that, you can incorporate in the feed, so that the feed will be very good protein, or you can get, instead of it will be utilized also, instead of wasting all this biofloc, at least some quality biofloc product you can take, it can be utilized for this one. But otherwise also, when you are harvesting and when water treatment protocol should be followed, and the maximum advantage you will get when you again you could reuse that the oxygen, deprived oxygen situation, if you remove, you can get. So we conduct some training program with so many farmers. We are working together, and then we are at the national level. You know, there is some lot of attention should be given for species wise. What should be the SOP, the kind of SOP required, and we don't have, even for shrimp, we have not defined the stocking density which should be allowed for the biofloc system. So that is a requirement of the sector policy, and instead of going for super intensive culture, you can go a moderate culture with the semi-floc mode of production also, and also the evaluating the biofloc and periphyton based system, the how to control that, and how this now the PMMSY is giving a lot of opportunity, and by that the capital, some subsidy also is given by the government. So it should be really at scientific side, it should be science-based culture. And really, it is a innovative technology which can give sustainable intensification, but we must all work for that. Thank you so much for the kind listening. And is there any question? You kindly go ahead.
Sir, okay, ma'am, you can ask them after that.
Okay, thank you. Actually, in Hyderabad, when I'm trying to do biofloc using borewell water, what are all the minerals I have to ensure that are available, because salt is less in borewell. I have to raise the salt PPT level for shrimp.
Okay.
So, what are the other minerals? Many minerals are not available in borewell water. So, what are the minerals that I have to ensure I add extra?
So actually, this is in the for shrimp, there is a requirement of if you are talking of shrimp, there is a requirement of this kind of Venamine, and the shrimps are some most of them are urihaline. We tell they can go from 0.51 ppt salinity to almost like 45-50 salinity. So they can adapt very well. But what we want when we are keeping them in very high density culture like bioflok, definitely you need to look for the mineral, and the mineral which are important is, but salinity you can go as low as 2 ppt, 3 ppt, and that is fine, because you have to see your environment also. You cannot just make the salinity 30 ppt, but when you exchange the water, that water should not create any hue and cry, because nearby there may be some paddy field and some other thing where the water should not do any salinization. So you can keep salinity only 2 to 3 ppt, that is all right. But the mineral wise, which are important is calcium, magnesium, and potassium, and of course there are sodium chloride and all these things. But these three are absolutely most important thing. And calcium, which is required, is like for 1 ppt of water, you need a calcium of almost like 10.6 ppm, and magnesium of 39, around 39.5 ppm of magnesium. It means ppm, you understand, that much gram per ton of water you need. And potassium also equally like 11, like that ppm is required. That is for 1 ppt water. If it should contain that much, but you should try to maintain a level of mineral is like 10 ppt water. It means calcium should be at least 110, and magnesium should be 390, and potassium should be around 116, or 110, 116. So that is the level you can maintain the mineral to get very successful without any problem the culture. But otherwise the PPT, the salinity, you need not take it forward more than that. Yeah.
And this sludge removal, doctor, you were mentioning, should it be done on a daily basis or any certain regular periods? How?
Sludge removal, it should be like, it depends on if you are in the beginning of your culture, you know there is no much turn and there is no much sludge generated. You need not do regularly. You may do after a week or after 10 days or after 1 month or so. It's fine. When it is, the animals are very small. This one, and so much of this one when you are putting into the system so much of carbohydrate and this one, then make sure that even at times after every meal, if you're see a lot of black particles and this loss is finally floating, and some smell you are getting, it is important to remove this loss, and this will it is difficult to remove from this system.
So once we remove the sludge, then again we need to add carbon and everything for the flock to form.
Actually, flock is which is floating, which is they immediately they settle in the bottom. So you just need to take out the loss from the bottom. They are not technically there, but if your level of flock is less, or if your ammonia is not still getting controlled, you can use the carbon nitrogen ratio and create more of a flock. Yes.
Doctor, can we get your phone number, doctor, if I want to call you?
Yes. My phone number is 90
yes
2573
573
94
94
99. Yes.
Thank you, doctor. Thank you so much. It was wonderful listening.
Sir,
Can you repeat your phone number?
902573 9499. 902573 9499. there may like all of yeah.
Sir, I have a question.
Yes, please.
Actually, what we see in our fish farmers,
Yeah.
but as you have said, they focus more on the training or showcasing their possible drawback behind this.
No, I didn't get. Can you please?
Actually, sir, what you have shared at the very beginning of your presentation, that many farmers or their all those things. Okay, and what this is a very true fact. They mainly practice fish farming, and those are mostly shady, babda or concerned upon, and the production is not up to the mark. That's why they are training or showcasing their farms. So, what can suggest something? Why the production level is not that up to the mark, or it's not that successful?
Actually, I will think differently. Actually, shrimp farming, if you see, it is very much successful by based aquaculture. And coming to the grow out culture, it is very, very capital start biofloc based shrimp, very, very with low budget. You can't start this one. As I told, aeration is required for a grow out big farm. I'm telling equally for a small unit like 10 ton capacity, people have achieved is only 100, 120 to 100, like that 130 kg. It means, and in a Indian system kind of thing, you can get up to 7-8 kg production for the nation. You cannot see this tilapia can be produced, even 100 kg people have produced for 1 ton, for 10 to be produced. So that density can be cultured, but shrimp that is inherent capacity you can get per ton of water. So maybe people are not very, very interested or convinced. They want to see in inside areas, and they are getting also. And shrimp is again one step forward. Coastal area, those we suggest them first to do one biofloc, one culture with fish under more optimal conditions is required for shrimp. That is one.
Dr. Panigiri,
Thank you.
can you suggest what are the other target fish which are successful?
Yeah, successfully is see this that is like the how you take it. What is your success criteria? Based on that, you can define your great success. Your fish should have a proper market value. And like other.
So my question is around what if biological is possible?
Yeah.
What about commercial? What are
That's coming to that.
Wow.
Actually, there are major two aspects. One aspect to create a very optimal environment where there is no stressors like ammonia, nitrate. All these species can get that advantage. But unless this species is able to building, then it is you are not getting the full advantage of that one. So in that, the singi, the Desi Magur, Pabda, and even this other, except that INC, we have not even named any other, like Amur Carp is a very good species, and gift tilapia and all, they are found and be there because they can utilize the biofloc effectively. But again, the market and other thing you have to decide that one, but value also. So that goes with that one. So those are all successfully people are doing this. At least the COI is very successfully in the biofloc system. In the fresh, and some of the brachystotle, which are also very good candidate species. Some of our farmers, they have tried that brachystotle species, but that can adjust in the fresh water, and they are very fast growing animal, and they can utilize the biofloc also. So we have seen they are very good small species also.
Sir, you mentioned Amur Carp and common carp. Do you have any examples, radiation level?
Yeah, I think they're getting good. I just compared with, you cannot like compare with this one. Like it will be like when you talk of this Rohu or the Katla, and their performance 300, 400 gram, beyond that they go 600 gram. They grow easily in the biofloc system. Some of the farmers they have grown, and they have that idea in 8-9 months time. That is what.
Sir, I am working in the area where the production.
Right.
Frequently, they are coming to the fishery extension officer for they are very interested to do the biofloc for the ornamental fishes. So, can you please provide me or tell me whether which economically viable according to the color and hand?
Yeah, right.
So which species are very viable or compatible for the biofloc?
I think that way, if you see one thing what you told, our experience also, that one, the texture, the color and all will be very bright. All the ornamental pieces, I can tell, it is only some ornamental species can utilize that one, the biofloc, they can utilize that and convert that into flesh. Some animal cannot. So that with our experience, what I can add to your query, that there is a way to culture ornamental fish breeder or this one, they need Moina or the Cladocyrans and all. They are very good species to be cultured in the live feeds using this biofloc, and of course this ornamental at the nursery stage, that they will be very immune and disease immune by using this one. Is that to pinpoint some species? I'm sorry, I cannot, because it is under trial. Many people are trying. Little data we have. Yeah. Any quick question?
Hello.
Good afternoon. Hello, sir. Hello. Yes. Good afternoon. Good afternoon.
Sir, audible?
Hello. Hello.
Sir, am I audible? Actually, how you restricted to that, that only by salt addition, only the salinity could be maintained in the shrimp farming, but till it to be observed that after maintaining salinity up to 350 or 2.5 to 350, the survival is too low. So you also added that all those minerals is to be missing if you can do soil addition. So sir, is there any way or any test kit by which we are available? We have our all those developed very low budget. Is it possible?
You can write to our institute. You can get the mineral testing kit.
Sir, I think
We had tried bio salt addition also.
Bio? Hello, please tell me.
Hello, sir.
Okay.
Hello, sir. I'm Sanjay Mukherjee. Hello.
Tell me.
In case Agur Singhi Pabda, which have naked scales, felt disease more than scales seen in catfishes, and how to get rid this if already disease has been diagnosed.
I think that is your observation is very, very correct. And if you are, like this is the scale is which the fish avoid all this kind of immediate response. But once in this biofloc, your biofloc system, mostly now is missing in case of freshwater is not very much well thought of. They just allow anybody anything to happen because the biosecurity aspect very strongly. So here, once your pathogen like parasite or then because of the high density, this catfish and the jacket skin thing, they will attain the disease, the tail rot, fin rot, and the like US kind of thing is on the kind of problem you are having. We have been freshwater this disease management control, and it is able to with some minimum. But what I can suggest is you should have a proper quarantine, like that little more one step above. You have to do proper quarantine, and particularly like if it is how many, like if it is a fungus, there are specific things. If you can identify with some expert, you can definitely treat it, but that. So we can privately talk also if you have some specific problem later, because we have.
Sir, I thank you extremely. It was an extremely well presentation. It is an excellent presentation. The regularly conducting the training on this thing on the biotop.
Thank you.
Thank you, sir. Again.
Okay.
Next presenter is waiting. Our next presentation is by Dr. P.K. Sadhu, and he is one of the excellent workers. He has excellent main topics, and one of them was albulas, control of the albulosis. He has worked on albulosis problem for so many years, so we requested them, sir, can you please take a class, and he agreed. Now he will give a lecture on albulosis. Dr. Sahu, sir? Yeah. Hello.
Hello.
Yeah, good afternoon.
Hello. Good afternoon. Is it
Yes, you're audible.
whether my slides are visible?
No.
Just 1 minute. I'm trying. Dr. Marna, could you please tell me why it is not coming?
Sir, go to present now. Then go to share full screen.
It is showing entire screen. I am
Share. Then okay.
I have put to entire screen.
Okay.
No, it is not showing okay. That's what.
Sir, select the screen. There will be a screenshot showing your
Already I
entire
have put share.
Kindly select that.
Is it coming?
Share. Share. Yeah. Now it's coming. Now you open the presentation directly. And kindly press the hide button just beside the stop sharing.
Excuse me. Why it is not coming?
Sir, you just open your PPT, sir. It will be shown automatically.
I have opened my PPT.
It's not showing. Your screen is coming. Yeah. Okay, sir.
Is it okay?
Now, sir, please speak. Yes, sir. No need to put the slide show, sir. You can move it manually also.
Okay, thank you.
And sir, please click the hide option. Just at the bottom of your screen, there will be a hide option. Please, beside stop sharing, there is a hide option. Please click on that.
Thank you. Thank you very much.
Perfect, sir.
Okay. Sorry for the trouble. Okay. Good afternoon, everybody. First of all, I must thank Dr. Das, Director CIFRI, Dr. Manna, the coordinator of this program, for giving me this opportunity to share my little experience, what I do have on this particular parasite with you people. Hope I am audible, clearly audible. I think it have been already covered. I will not repeat all those things. We have a large number of bacterial problems, viral problems, fungal problems, and parasitic problems in freshwater aquaculture particularly. And we'll be speaking only on this parasite arglosis, which you are seeing, which is a major survey program throughout Orissa and Andhra Pradesh particularly. And if you see the figure, it is not differing to any of the states. And also we have conducted a massive survey during 2008 to 12 also. So if you see the result of those survey, so every alternate pond in the system is mostly being infected with this particular parasite in a culture system, freshwater fish culture system. And this particular survey, which has been conducted under the National Surveillance Program during 2014 to 18, we found parasitic infections are much more dominant in the culture system, besides the bacterial and other infection systems. So around 67% infections are parasitic infections. They cause a massive damage, like Argulus, which is a major killer, and Dactylogyrus, that is gill flu, which is also equally important parasite in the freshwater aquaculture system. And Argulus, the prevalence percentage are around 20%, but if you see the one more triangle in this area, 37% infections are mixed parasitic. In this mixed parasitic infection cases, mostly also you will find there is presence of Argulus. So Argulus seems to be a major challenge to the freshwater aquaculture system. Similar to Argulus, we call it as a fish lice. In the sea also, in the marine environment and also brackish water environment, there is another parasite similar to freshwater fish. This is Argulus species, that is Lepidoptera species, mostly affect salmons and other fin fishes. And also there is one Caligus species. There is a huge institute in Europe, particularly in Norway, which is working on the sea lice, particularly. They are addressing only sea lice infection, sea lice research center, and because they are losing more than 300 million euro per year because of this sea lice infection in salmon farming. So coming to this particular parasite, you can see this particular rohu, which is a live rohu, and it is loaded with parasites. So how the fish will survive, you can well imagine how important this particular parasite is. So if you think this particular parasite is an obligatory parasite and obligatory ectoparasite. Obligatory means it requires its food from the host. Without food from host, it will not survive. And it is a generalist parasite and mostly affect many fish species which is having scales. Most of the fish species having scales are being infected with this particular parasite. And they are all micro predators. So they graze on the mucus, on the skin surface. They cause a lot of irritation. They feed on mucus. They inject some toxins like proteases and other anticoagulants, where at the place of attachment, at the place of injection, there will be fine hemorrhages will appear because they are injecting anticoagulants. So bleeding will be continuously there. They will eat on the blood and they inject. So they eat on the dead tissue. They eat on the mucus. So they depend upon the host for their survival. And they are ubiquitous in distribution. They are found throughout the world except Antarctica. It has not been reported. Otherwise, this particular parasite has been reported throughout the world. And this is one of the most important parasites in freshwater aquaculture system. If you want to control this particular parasite, you must have some basic knowledge on this biology of this particular parasite. These mature male and female parasites, they meet after mating. The female carries the eggs. When it carries the mature eggs, they leave the host and they search for how the eggs are being laid in process means in groups. The eggs will be laid, and once the eggs is laid, it is covered with a hard coating, just like our like your aldehyde, glutaraldehyde type or chemical type of coating will be there, which is very difficult to be damaged by any chemical substances. So it will be, the eggs will be in chain. One single female can lay eggs in one clutch of more than 150 to 200 eggs, and it varies from species to species of Argulus. Because within Argulus there are more than 129 valid species of Argulus are there. I am not going into all those details of those species available. We will focus more on this Argulus biology and what they are doing and how they are being managed in the system. Because I came to know that most of you are mostly heterogeneous groups. Some are students, some are farmers. So that's why I will say. So once the eggs are laid on the hard surface, they will take some time to develop into embryonated eggs, and it varies from species to species, and it depends upon many factors, including many environmental factors, particularly temperature, which modulate the hatching of these eggs. At low temperature, it may stay even up to years without hatching, and it depends late. So accordingly, the hatching period also varies. Sometimes the species which are prevalent in Indian waters, mostly in the tropical environment, they take 18 to 20 days to hatch. Sometimes it even takes 6 months to hatch. Once the hatching occurs, the metanuclei will come out from the egg. Again, the metanuclei should get the host within 4 to 6 days, otherwise they will die. And that also depends upon how many days the one metanuclei can survive, depending upon the species of the parasite. So they find out a suitable host And again, once they are attached on the host, they will start again feeding through their different mouth parts. And it is very easy to identify whether the infection has come up in a particular pond or not without catching the fish. The very easy way is to fix a bamboo pole in a pond and lift it after 24 hours or 48 hours. You can see the clutches of eggs. These are the egg clutches given by the parasites. Even some ideas are there. If you fix a pond in a particular pond, if you are putting IMC in the culture system, you will find on the top of the bamboo pole, if it is infected, you will find the eggs. Mostly you will find the cattle species is infected. At the bottom, it will be infected which prefer roe. Some species they prefer, some strains within the species also they prefer mrigal. Some species they infect mostly catla. We have seen silver carps are being infected, but roe, catla, mrigal are not infected in the same pond with the same species of parasite, but there is variation in the strain. And you can see one photograph which I am showing snails. These are living snails and the eggs are being laid. So the eggs are laid on any hard surface, including the vegetations which are on the bottom of the pond or on the sides of the pond dikes. And you can act as a biological transmission or vector for the transmission of eggs. And it can go during netting or any operation from one pond to another pond or another water body easily through this, and transmission occurs very easily. You can see these water weeds having laid, the eggs are being laid on these water weeds. The clutches, you can see the clay clutches. So if you leave these plants also, you can see, you can go to any pond and lift it. If you can see if the eggs are there, if fish infection is there, you can suspect there is infection. So you have to think for its prevention or control. And coming to the anatomy of this parasite, these parasites mostly feed with they cause a lot of irritation through their maxillary suckers and feed out through their proboscis. And you can see the very well the female parasite carrying the eggs and the male parasites. And species differentiation is very difficult because there are more than 129 valid species are there. So it is quite difficult. We have to develop some sorts of markers. We have developed for whatever species are available in India. We have developed some molecular markers which can be easily identified. These parasite species can be identified. So during our survey, we have found out if there is infection, more than 29,000 loss per hectare per year in a particular water body occurs due to this particular parasitic infection. And on an average, 48% of the water bodies of the carp culture are being affected in a particular period. And we have estimated the loss. This is the only disease for in freshwater sector which has been, for which loss has been estimated, that is, it goes up to 300 crore. So the loss might have been increased much more because of this single particular parasite, which is causing huge loss to our freshwater aquaculture system. If you see the loss is not because of the mortality. Mortality loss is very less. Because of this parasite, there will be less mortality. But the mortality will drastically increase because this parasite, they suck blood, they cause a lot of irritation, they inject anticoagulants, what I was mentioning. Besides that, they also inject some sorts of toxins which suppress the immune system of the fish. Once the immune system is suppressed, so all kind of infections you can expect, the host will be infected with number of parasites or bacteria because of this parasitic infection. But in experimental condition or in a experimental pond condition where we have produced and or in natural cases also, most of the cases are using going for application of drugs, that is also very less. But the problem is this parasite causes huge loss in growth of the fish. Every day, if you think of carp farming, the Indian major carps, they on an average they should grow at the rate of 3 gram in a standard culture operation. 3 gram they should get a growth on an average 100 gram they will grow in a month, if you see. So what happens once these parasites happen at the early stage, when they are hatch out to metaloclase stage, till 5 to 10 days, you cannot see those parasites in a naked eye. So even if you are catching the fish, you are doing cast netting and examining the fish, you may not able to see the parasite. The parasites may easily escape from your notice. So they will be causing huge irritation. So the fish will be moving here and there, and scrubbing its skin to the hard surface, and it will not take any kind of natural feed at that or artificial feed. It will reduce its feed intake. So what will happen? So, and because of the irritation, because of the blood loss, because of this feeding of this mucus contents, so there will be drastically growth reduction. The fish will not eat, and simultaneously the feed which you are offering every day, so there will be reduction in the feed to growth will be much more. And you can see at the initial stage you may find focal hemorrhages at the base of the fins or on the body surface. There will be mild hemorrhages, but parasite number is too high. But they will find mild hemorrhages, and it will vary from even species to species the type of lesions. Even in some species, you can see massive hemorrhages. Small number of parasites, but massive hemorrhages. Massive hemorrhages at the caudal fin, at the base of the fin, or all kinds of even tails. Also, you will find large number of hemorrhagic areas, focal ulcers, and sometimes it may lead to large-scale dermal ulcers also. It will be there. It will expose the fish to the secondary invaders like your bacteria or other kind of parasites, because not only it causes injury, also it suppresses the immune system of the fish. I am not going into all those details. So how their immune system is modulated, we have done a lot of this. At the look, once it is attached, at the site of attachment, it causes inflammation. So inflammation means swelling of this. Our interesting observation, I will just mention. You may ask why, some of you might have seen this, particularly the healthy fishes. Healthy fishes or adult fishes or adult or juvenile fishes, they are being mostly infected. You will find small sized fish like your fingerlings or early juveniles. They include several factors, particularly if you will find in case of calves, the inflammation is much more prominent in case of young stages of fish. And inflammation serves as a healing agent or curing agent or a repelling agent for this particular parasite, so that they can be easily detached. So they will not be much more infected when we have kept all kinds of species of different sizes. We have looked into how the inflammation is governing this particular parasitic infection. Again, within the species, there are a lot of variation. If I have slides, I can show how roe is much more preferred species for this particular parasite, followed by catla and others, minor carps. Then comes your grass carp, which seems to be much more resistant within the carp species to this particular infection. And once there is attachment, there will be hemorrhages, there will be infiltration of large number of inflammatory cells, and there will be increased number of phagocytes. There will be, you can see one slide, histological slides of it, and how these blood cells are being migrated to the site of infection, how there is massive hyperplasia in the skin itself. Okay, so I am not going into the details because the time is very short. I have been given only half an hour. So what I will suggest here, so this particular parasite causes not only inflammation, not only hemorrhages, at the initial stages causes huge amount of mucus production, because the fish will try to get rid of this particular infection. So slowly they will eat on more amount of mucus, and slowly you will find large number of parasites with less amount of mucus on the body of the fish, and they will damage to all the caudal peduncles or the fins, and they will be also infecting or attaching to the gill operculum also, and sometimes the fish can die with few number of parasites. sometimes the fish can survive with large number of parasites, which I was showing on my earlier slide. A single fish with thousands of parasites, but it was surviving in the pond. So ultimately, how we have to think of prevention. So prevention, most important thing, mostly people are controlling through chemical treatments. All kinds of different kinds of drugs and chemicals are being used currently to treat. But if you see, most of the drugs or chemicals which are being used are not able to fully control this particular parasitic infections. So couple of institutes are working, even CIFRI is working, CIFRI is working, and CIFRI is working on this particular parasites. We are struggling with these parasites since last 15-20 years to come up with. You can ask why you will go for vaccine strategy. We are thinking of long-term protection throughout the culture period. That's why we are thinking. And in the absence of any kind of permanent control program, so we have developed some kind of comprehensive strategy with the available drugs, how you can go for treating. Because these parasites, the people, the farmers, whatever drugs they are using, either they do not know the optimum application dose, that is dose or time schedule or the duration of giving the drug to the fish, and how much requirement is there, when it should be given, at what temperature, how much dose should be given. So nobody is aware. Even we are not aware about many drugs. But still we are trying to develop the management strategy, how the farmers can easily get rid of this particular problem. So main thing is, if you want to get rid of this particular problem, we will give you some strategy, like these argus, they reproduce around the year. So you have to develop on this some control strategies which should get rid of even from the pond, when of any culture pond in the month of April or May or June. So what we would suggest, so you have to treat that pond with some kind of drug. There will be X, Y, Z kind of drug. Why I am telling different kinds of drugs, I am classifying the drugs based on the mode of action. Most of the parasitic side compounds, they cause hyperpolarization of the nerve endings or hypopolarization of the nerve endings of the parasites. That means, suppose for example, some drug like ivermectin, which is also widely used, or methrin, delta methrin or cypermethrin kinds of drugs, they are being mostly used. So suppose delta methrin, they cause excitation of the nerve endings. Suppose the ivermectin, they carry ions, so that the parasite will be easily paralyzed and died. And there are some drugs which are also able to prevent the molting of the parasite, because this is a crustacean ectoparasite. It needs frequent molting during its developmental process. If you want to stop its molting, there are some drugs that are used. I will show you those drugs which can be used in the pond system and it can prevent the molting. So what I want to emphasize here, so because most of the time we are empirically using some drug continuously in a culture system. Like people are using cypermethrin or deltamethrin or ivermectin group of drugs continuously in a culture system. These parasites, like any other parasites, or like your malarial parasite in human being also, they are developing drug resistance. This parasite is also developing frequent or inappropriate use of drugs in the culture system. We do not know what is the amount of drugs should be used at a particular time in a particular point. So that makes the major problem with these parasites. So they are developing slowly resistance. So I will tell, for example, in a pond system, when you are applying drug, these parasites have a strong sensory power. So in a pond, you cannot immediately apply throughout the pond. The drug cannot be applied. So once you realize there is something, danger is coming to it and it may die. So in a particular time point in the pond, you will get mature parasite, you will get immature parasite, you will get the metanuclei, and you will get some eggs. So what will happen? Once you are applying the drugs, the fish will be moving from one side to another side, and the parasite, if it is supposed to lay the eggs maybe tomorrow, it will leave the host and it will go and lay the eggs immediately. And most of the chemicals of the drugs available are not able to kill the eggs. They are mostly targeting either the adult parasite or the juveniles, because the eggs are very sticky, very hard, and there is no such chemicals available which can easily kill the eggs. The eggs are very hard, very hard. So that's why once you are applying the drug at one side of the pond, the parasites are laying eggs, and the eggs will be hatching maybe after 3 weeks or 4 weeks or 6 months, depending upon the environment, depending upon the temperature. So it will hatch and it will take certain time. So what will happen when by that time it might have been degraded, and again the eggs will be hatching and it will be releasing metanoply. Again after a few days the infection will come up. Again we will blame that the doctor who has prescribed it has not properly prescribed the drug. So you have to understand the biology of the parasite. So accordingly you have to go for the management strategy by using different drugs. Now people are also using cascade of parasite control strategies, like they are employing different biological control methods. Different fish are available, like in case of salmon lice, there are a couple of fish species which can easily eat those sea lice because they are quite huge. And here also people are using some guppies, but it's not that much acting as a biological control system. But our farmers are doing, they are putting some sorts of bamboo sticks or bamboo mats to remove the eggs, whatever being laid. They are drying it for a few weeks on the sun. Again, they are putting and they are removing the eggs. Again, they are putting and they are removing the eggs. So it is very difficult to get rid of those parasites. So that's why you have to think or understand properly the transmission cycle, the biology of this particular parasite. If you can think, then you can able to control to some extent. Even says I was vector system. You can while netting, they will come to the surface. Again, they will move to the another pond. They can easily spread the infection. So what to use to suggest in every culture system, even there is parasite or not, if you apply the drugs are very cheap. You can apply one kind of drug like the chitin inhibitors after 3 months after 6 months of culture. You can apply another mode of action of drugs like ivermectin group of drugs or deltamethrin group of drugs. You can apply to control whole culture period of 9 to 10 months or 11 or 12 months of culture if you are doing. So if you will circle this drug or do the different mechanism of production of drugs, so you can able to, otherwise it is very difficult to get rid of this particular parasite. Just like any other disease, any disease which is under stress or with deprived nutrition, or if you are going for high stocking densities, or there is even temperature fluctuations are there. These are the factors which mostly influence for any kind of pathogens, any kind of parasites to cause infection easily. So what do you like to suggest? You prepare the pond with 1 dose of parasiticide application, and again get the seed when you are getting the juveniles or the fingerlings. You get the seed from a pond. You insist to get the seed from the treated pond also similarly, so that the parasite will not come through the fish and your pond is free. So that at least up to 6 months you can maintain a free culture period of 6 months without this particular parasitic infection. And monitor regularly. Every month you have to go for castrating so that you can find out whether this parasite has come up or not. That is the very easy way. Or else you can fix a bamboo pole, what I was telling. So you can also look into the eggs. If there are adult parasites are there, they will lay eggs. After 2 days or 1 day, even you can find the eggs on these hard surfaces easily. And you have to rotate the chemicals, whatever chemicals you are going to use for particular infection. I will not go into all those details. There are a couple of drugs are available. I will not go into which can be used at weekly interval for 5 weeks. It can give even full culture period of 6 to 8 months free of parasite. This drug, lopinirole. Like if some drugs like teflubenzuron, like drugs which prevents molting of the parasites, or diflubenzuron can be used, or like ivermectin group of drugs can be used, which are most commonly used and which has very less detrimental effect. Like ivermectin group of drugs, it can stay in the system within up to 17 to 20 days. So by that time, till that period, it cannot, the parasite infection will not occur. Even some parasites will be living, even they will be attached after your ivermectin group of drug application. Mostly it is applied for 3 to 5 days depending upon the grades of infection or the amount of infection persist, either through feed or through by water application. You can apply both the ivermectin group of drugs also. Or if you are going for like deltamethrin group of drugs, deltamethrin is nothing but your methrin group of drugs which has these both transacting substances, and it is methrin means it is derived from a methrin plant, which is the natural substances, natural derivative is like most closely similar natural derivative is deltamethrin. Just one second. Deltamethrin group of drugs that can be used. That deltamethrin group of drugs, if you at all you are going to use, it is highly metabolizable and very easily degraded from the aquatic environment. So what you have to do, you have to go for repeated application depending upon the temperature. If it is winter, you go at 10 days interval, or if it is summer, you go at weekly interval. But minimum 3 applications you have to give, because after the first application, most of the living parasites will die. After few days, if some eggs are laid earlier, they will hatch after 7 days. This deltamethrin will degrade within 3 days from the water environment. So if again some eggs, whatever being laid earlier, they will hatch after feeding. So you have to know the drugs, how to apply, what is the literatures are available. I will not go into all those deeps. You might be feeling hungry. I am taking too much of time. So what I want to say, we have developed one Android mobile app. It is known as Treat My Fish app. It is available in 3 languages at present: English, Hindi, and Odia. So it is an interactive app. You can go through this app. You can download from any Android mobile. You can easily download from Google Play Store, and where you will find about information about different diseases in freshwater aquaculture system. The interesting part is there is one place where you can report disease, and you will find it will ask all these details of your pond. What is the water depth? What type of species you are culturing? What drugs you have used? And you can also upload 3 different photos. So it is not only for argillosis, any disease problem, you can upload the photos, you can send us, and we can immediately reply you back. But sometimes, of course, sometimes if it is not at all working, you can directly also contact me, directly disturb me, or you can send the photographs through WhatsApp, and we will immediately come back to you. So this is in gist about this argillosis as well as this control mechanism. I have not gone up to the detailed science part of this, but if you understand the biology of this particular parasite, you can easily control it. So thank you very much.
Excuse me, sir. Sir, recently in this month of March, after diagnosis the Argil-Chobis Porgona, I have applied Albonex FX. Its composition is albendazole and invermectin with and I have applied Kemanofol before applying this. The dose was 5 grams of Albonex FX for 1 ton of live fish. Ma'am, sir. But after 6 days of application, only Katla species swims abnormally as and daily 3 to 4 Katla dies like this. I have seen there is no parasite on the body it happens.
Okay. If you see, most of the drugs which are being used in agriculture, they are not being or being used. They are mostly derived from the animal science aspects. So, truly speaking, they are not being properly studied. So, the internal parasites, you will get rid of the internal parasites. And it contains some emamectin group of drugs like your ivermectin, which belongs to emamectin, for causing paralysis in the parasite, so that parasite will be detached. But that drug also, it is simultaneous, it will cause hyperpigmentation and stress is even sometimes it leads to mortality. So I do not know what they are dying continuously until you examine it. It is very difficult to tell. If the if they are being hyperpigmented means they are, if it is blackened or pigmented, too much pigmented, probably you have to go for reduced dose level of from you. So we have to examine if there is any other factors are responsible for the mortality.
I have seen the Katla species operculum. The color is becoming violet, bluish, greenish type like this.
So I think you are from which place?
West Bengal.
No, West Bengal.
South Chobbish Pargana. South Chobbish Pargana.
So why don't you say diagnostic centers, or you can take help of Dr. Mana and others also, so that it will be easy to advise you why what exactly is the arbutus infection. The fish might be dying, one or two like dying. So once you apply the drugs also, it acts as some kind of, they will die much more. But there are many factors in the pond system which is very difficult to say at this stage without examining. I will not be able to justify.
Any more question? Sir, if there is no question, so yeah.
Now,
Please.
hello.
Yeah,
Are you able to hear me? Yeah, see, to prevent these infections or these parasites, so what all natural chemicals or natural products can be used? Like we say,
Actually, carbonate, if you will go for a natural product, I do not know, most of the products are ineffective. You have to depend upon the parasites. These parasites, even if you are maintaining optimum water quality, these parasites will also, of water quality, the instance will be less. That we have seen. So you have to maintain optimum pH of 7.5. The incidence will be less. Or else, even if there is go for just lime application, what you are mentioning, so that will give some kind of soothing effect to this post effect, but it will not detach all the parasites. It will improve the water quality so that the fish will be under less stressful conditions. That's why.
thank Dr. Sahu sir. Despite his sudden illness, was able to give his lecture, and thank you for this excellent presentation. These are not very exhaustive, but very, very useful. I know that the harbors is the major problem. So thank you, sir, for your guidance to all the participants and those who are seeing YouTube. And if anybody wants to please contact the CIFRI of him directly about the argulas. He is the best man about this. Thank you. Okay. Now,
Thank you, Dr. Mana. Thank you very much for giving me this opportunity once again, and thank you, CP whole team. Thank you.
Thank you, sir.
Thank you.
Now it is 1:30. So we will again at 2:30. We will join at 2:30.
Join at